My dear students, greetings. In last class, I tried to explain what is linkage, what are linked genes, what is a linkage group, what is the significance of the linkage. Now, in this session, let us try to understand the crossing over and recombination that is included in class 12th, unit 7th. In your syllabus, it is simply given as a recombination. But recombination is because of crossing over. So in linkage, what we have said, the two genes on the same chromosome, if they are very close, there will be linkage. If they are located away from each other, they are non-linked genes. For example, here you see there will be linkage between these two, there will be no linkage between these two. All the genes that are located in a linear fashion on a single chromosome is known as linkage group. The concept of linkage states that the different genes present on the same chromosome are linked and inherited together. First generation, second generation, as a unit, as if they are a single gene between the generations. However, linkage is not a rule. It is not a rule that the gene should be transmitted from generation to generation as a unit, as a linked one. Some exceptions are observed later on after Mendel, and the progeny may show new combinations of characters in low frequency. The parental characters get mixed or they are transferred in first generation or second. Thus, linked genes to get separated and make new combinations by a mechanism called crossing over. So, if you go back, for example, if you see, these need not go linked as a unit. They may get separated because the distance between them is more. The concept of crossing over was first proposed by Morgan and his student Casil in 1912. T.H. Morgan while working on Drosophila. What are the salient features of the crossing over? Crossing over is an alternative linkage. If there is no linkage, there will be crossing over. As the distance between the two genes increases, the possibility of linkage decreases, possibility of crossing over increases. They tend to get separated. It separates the linked genes on the same chromosome. The two genes present on the same chromosome get separated. It takes place through the exchange of corresponding parts of non-sister chromatids. This point you have to remember. Homologous chromosomes, chromatids in each chromosome, 2 chromatids total. During the synapsis, 4 chromatids. Exchange of chromatid pieces between non-sister chromatids. This is very, very important. Of homologous chromosomes. What are homologous chromosomes? Similar chromosomes, a pair of chromosomes, one comes from the male parent, another comes from the female parent. Similar in length, similar in centrosome position, similar in gene sequence, similar in gene number. They are called homologous chromosomes. Recombination of genes, a universal occurrence. It has been observed in almost all the species of plants, animals, microorganisms. The genes show a tendency to enter the gametes. The genes show that gametes in combinations frequency than parental. Crossing over depends on the distance between the genes. This I am emphasizing very frequently. As the distance between the two genes increases, the chances of crossing over increase. More the distance, the chances of crossing over will be more. Genetic variations in the populations are mostly due to crossing over. If there is linkage, the population will be similar, offspring will be similar. If there is crossing over, there will be variation. One of the reasons for the variations is crossing over. One of the reasons for another is mutations. There are two reasons for variations in the population. Main reasons, genetically speaking. One is crossing over, another mutations. So this no-linked genes, possibility of crossing over between these two genes is more. Here very less because they are very close to each other. Then mechanism of crossing over. How does the crossing over take place? Between chromosomes occur during meiosis cell divisions during Pachytene stage of prophase 1. Where this crossing over takes place? In meiosis. In meiosis 1, meiosis 2. In meiosis 1, prophase. Again in prophase, leptotene, zygotene, pachytene, diplotene, diakinesis. Where actually the crossing over takes place? Prophase 1, meiosis 1, pachytene. It is also important, very frequently asked. Then it occurs in all sexually reproducing organisms. In all sexually plants and animals. It is called meiotic crossing over because this crossing over takes place during meiosis, meiotic crossing. Now, there are some crossing overs during the mitosis also, mitotic crossing overs. So, we are not going into those details at this stage. Crossing over is responsible for genetic variations among populations. Entire crossing over process is completed in two stages. Synapsis. What is synapsis? Go back to meiosis. Meiosis 1, prophase 1. In prophase 1, what are the stages? Leptotene, zygotene, pachytene, diplotene, diakinesis. Second stage, zygotene, the two homologous chromosomes come together, stand side by side. That is called synapsis. The two one from the male parent, another from the female parent come together, stand by side. Then crossing over in pachytene. Synapsis in zygotene. Crossing over in pachytene. So this is stages of crossing over. You can look at here, A, B, this is another homologous chromosome. Here A, B, dominant genes, A, B, recessive genes, homologous chromosomes because the two chromosomes are similar. Look at here, this is the stage in zygotene. Now in pachytene, you can see here, this is crossing over, this X-shaped structure is called chiasma. Here exchange takes place between non-sister chromatids. These two are sister, these two are non-sister. They belong to two different chromosomes. These two are sister, but these two are non-sister. Exchange of chromatids, pieces, fragments between non-sister chromatids is crossing. Now, after crossing over, the situation is like this. This is normal, but this has got a new piece, small b. This has got small a. This is in diplotin. Then here you can see chromosomal after separation, crossing over of chromosomes during the anaphase 1. So this is the mechanism of simple crossing over. Theories of crossing over, several theories have been proposed from time to time. Precocity theory, copy choice theory, breakage and reunion theory. So, precocity theory proposed by C.D. Darlington, in this, what this theory says, the two non-sister chromatids coil around each other and exchange the unwinding, here chromosomes duplication and coiling of sister chromatids, unwinding of sister chromosomes and chromatid breakage, reunion, exchange. So this chiasma, this exchange point is called chiasma, X-shaped structure. Second case, copy choice. So this is predetermined, almost predetermined here. The exchange here, look at here interestingly, not between these two, but between these two. Between these two, not between these two. That means there is a choice of chromatids for exchange or for crossing over. So this is shown in detail. Then breakage and reunion. According to this theory proposed by Stern and Houghton, 69, before crossing over, the chromatids break at a particular point. Here, look at here. Then they are exchanged, united. This is called breakage and reunion. The two chromatids break at a particular point, exchange the chromatid fragments and reunite. Then hybrid DNA holiday model. This is similar to this, but only thing is at molecular level, DNA level. There it is chromosomal level, it is a DNA level. At a molecular biology level, this is a very important aspect, holiday model. Here you can see this exchange points. Then what happens? Sometimes what happens, the new strands are synthesized. No recombination here, but here recombination. So this explains how the crossing over takes place at molecular level, at DNA level. Kinds of crossing over. There may be a single cross or double cross, two-strand double cross, three-strand double cross, four-strand double cross, multiple cross, even number, odd number. The crossing over may be just at one place or two places or three places, like that. Cytological evidences. At the time Morgan worked, he was not knowing what are actually chromosomes, and he was not knowing the cycle. Concept of crossing over was proposed by Morgan in 1914. However, cytological evidence came and evidence was not demonstrated until 1931. Kurt Stern, 31, while working on Drosophila, and Creighton and Barbara McClintock, 1931, while working on maize, designated they have planned some experiments to prove the cytological evidence of crossing over after almost 20 years. Significance of crossing over. Evolution, simply speaking, evolution, variation in the progeny, because there will be exchange of genes, exchange of chromosomes. Provides a scope for the study of nature and function of mechanism of genes, how the genes are inherited. Result in new combination characters which may be advantageous to the offspring. So new variations appear, new varieties or new offsprings. So this variation may be advantageous sometimes. Later we study inbreeding, outbreeding, like that. The variations caused by crossing over lead to evolution of species. New species. Within the species there will be variations, varieties. Chromosomal map, linkage map, genetic map, these are almost same. Chromosomal map means how many genes present on the chromosome, what is the sequence, which genes are linked, which genes are non-linked. Similarly, linkage map, same. If the two genes are there, whether there is linkage or not. Then genetic map. Concept of linkage crossing over states that genes are located at specific locus on the chromosome in linear order. Linked genes are inherited as a unit. The strength of the linkage depends upon the narrowness between the two genes, closeness between the two genes. Linked genes get separated by crossing over, resulting in recombinations. So here one point, suppose there are two genes close to each other. There are two genes that are far away. The crossing over will be less between the close genes, more between the genes which are located far away from each other. As the distance increases, crossing over chances increase. The above observations led to the idea of construction of linkage map. Sturtevant and Morgan, 31, for the first time prepared linkage map by showing 5 genes on X chromosome of Drosophila with the relative positions. On X chromosome of Drosophila, what are the genes? What is the linkage between the genes? They made use of recombination frequency to determine the distance between the genes. Suppose A, B, C. What is the distance between A and B, between the B and C, between the A and C? By making recombination frequencies. A chromosome map is defined as the graphic representation of genes. A diagrammatic representation of genes on the chromosome, on the same chromosome, showing the distance between the different genes of a linkage group. What is the distance between A, B, C, D, E? Between this is called chromosomal map. Map units or Morgan units. Map units or Morgan units. 1% crossing over is equal to 1 map unit. 1% crossing. For example, let us take 4032 and 4008 parental combinations appeared in F2 generation. In a cross 4032 4085 parental, but recombinations are 149 152 crossovers. So this works, this is 3.6 in total count of the FT generation, 96.4, just like parents. 3.6, the two parental characters mixed. So therefore the map units are 3.6. That is, the two genes are located on the chromosome 3.6 map units away from each other. So suppose this is A gene, this is gene, 3.6 map units. This is how they calculate the map units. So this is the linkage map or chromosome map of the Drosophila, 4 pairs of chromosomes. So this is a prokaryotic chromosome map, different genes, their locations based upon the recombination frequencies. Molecular recombinations at molecular level, not at chromosomal level, at molecular level. Genetic recombination or genetic reshuffle is the exchange of genetic material between different organisms, which leads to the production of offsprings with combination of traits that differ from the found in either of the parents. Those characters which are not found in the parents. In both prokaryotes, eukaryotes, best studied in bacteria, archaea, yeast, and phages, mediated by breakage and joining of DNA strand. 4 types: general or homologous recombinations, illegitimate or non-homologous recombinations, site-specific recombination, replicative recombination. These are different. Recombinations between the homologous chromosomes, recombination between non-homologous chromosomes, illegitimate. Site-specific at a particular location, the recombinations take place in phage, lambda phage. Replicative recombinations, particularly it is found in transposons with respect to transposon. Homologous recombinations, then illegitimate non-homologous recombinations, where large-scale sequence similarity apparent, no large-scale sequence apparent, translocations between different chromosomes, deletions that remove several genes along the chromosome. Recombination between two similar genes that are several million base pairs apart can lead to deletion of the intervening genes in some cells. Remember, it is between non-homologous chromosomes. So one homologous here, another homologous here, away from each other, but still recombinations take place between these two. Genetically, gene-wise, they are quite different. Then site-specific, a particular site, for example, in lambda 12 to 24 base pairs, at that place, what happens? The phage genome enters into the bacterial genome, lysogenic cycle. Site-specific recombinations require special enzyme machinery. Basically, enzyme system, integrase enzyme, there will be no enzyme known as integrase that integrates the phage genome into the host genome, bacterial genome. Good examples are systems integration of bacteriophage such as lambda into bacterial chromosome and rearrangement of immunoglobulin genes in vertebrate animals. These two examples, one lambda phage bacteria example, second is immunoglobulin genes. Replicative recombinations, transposons, jumping genes. They move from one place to another place. within the genome, sometimes between the two genomes, two different genomes. Replicative. Recombination frequency. We can use the frequency of recombination events between two genes, their degree of genetic linkage, to estimate their relative distance apart on the chromosome. So they say here, bacterial also recombinations take place through well-known, well-confirmed, well-established process, transformation, Griffith, Diplococcus pneumoniae, transduction, then conjugation, Lederberg and Tatum, here Gender and Lederberg. This is mediated by phage, conjugation tube here. What is the benefit of calculation the recombination frequency? So several, genetically or otherwise also there are several. One way to recombination frequencies have been used historically is to build linkage maps, chromosomal maps based on the recombination frequencies. Recombination frequency is not a direct measure of how physically far apart genes on chromosomes. However, it provides an estimate or approximation of physical distance. Correct, not correct distance, but approximately we can say A, B, C. We can say A is close to B, B is close to C. We can also say whether closeness is between A, B or B, C. Hence, we can say that pair of genes within larger recombination frequency are likely further apart, while a pair with smaller recombination frequency are likely closed. This I have explained. Thank you. This is what is crossing over, very important aspect in genetics. I think after the Mendel's laws of inheritance. Linkage and crossing over, I compare just like the two faces of the coin. When you see one face of the coin, you cannot see the other face. If you see the head, you cannot see the tail. And reverse is true. Similarly, when there is linkage, no crossing. Thank you.